Assalamu alaikum warahmatullahi wabarakatuh. Dear students, this is chapters the eye and the ear included in the neuroscience module 205. First, we're going to take the eye. In the eye, we're going to take microscopic slide, low power eyelid, high power cornea, and retina, as well as the data shoot will be eyelid, cornea, and retina. Here, this photo. We know that the eye is formed of three coats. The outer fibrous coat is formed of the cornea, anterior 1/6 and the posterior 5/6 sclera. Then we have the vascular coat, the iris, ciliary body choroid, and the nervous coat, the retina. And here it's covered with the eyelid. We have here section in the eyelid, structure covered by skin. In curriculum, we have 4 structure covered by skin: eyelid, lip, mammary gland, and pins. Okay, here is the skin covered with keratinized stratified squamous epithelium, and here are the hair follicle. And the other side is palpebral conjunctiva, stratified columnar epithelium with goblet cell. Between them we have muscle known as the orbicularis oculi and other muscle known as levator palpebrae superioris, which are skeletal muscle. Embedded in the torsus, gland known as meibomian gland. Between the skin and the palpebral conjunctiva, there is lid margin. This lid margin contains the TS sections here of the hair follicle. In order to differentiate the eyelid from the lip, look at this margin between the skin and the mucous membrane. If you see hair follicle, so it is eyelid. If you do not see, it is lip. Here, dear students, slide microscopic picture of the eyelid. One side skin, which is number 1. Number 2, the palpebral conjunctiva. Number 3, I can see here long hair follicle, which is the eyelash. Number 4 is the muscle, orbicularis oculi. And number 6 is the torsus, which is dense connective tissue, embedded at it the meibomian gland. High power slide under microscope and in the data show the cornea. As you can see here, the cornea is formed of 5 layers: epithelium, membrane, and in between it, stroma formed of substantia propria. The epithelium is non-keratinized stratified squamous epithelium resting on basement membrane, a membrane non-cellular known as the Bowman's membrane. And here is the stroma formed of regular collagen fibers parallel, between them cells known as keratocytes. And here is the descemet membrane, which is the basement membrane for the cell, which is known as the endothelium, flat cell with flat nuclei. Here another photos for the cornea: epithelium, Bowman's membrane, substantia propria or stroma, descemet membrane, and endothelium. The same: epithelium, Bowman's membrane, substantia propria, descemet membrane, and endothelium. Here is a section showing the 3 coats posteriorly: the sclera, formed of irregular collagen bundles to withstand tension at different direction as the extraocular muscles attached to it. Here is a vascular coat, which is the choroid, pigmented vascular coat here. And here is the retina formed of its layers that we're going to discuss now. Here is the retina. This is the sclera. This is the choroid. So we'll start the retina by the pigmented epithelium, rods and cones, outer limiting membrane, nuclei of the cells of the rods and the cones, which is the outer nuclear layer, the axons of the rods and cones, make a reticulum or plexiform or synapse with the dendrites of the cells of the inner nuclear layer. The cells present in the inner nuclear layer are the bipolar cells, the molar cells, amacrine, and horizontal cell. Axons of these cells will dendrite. The axons of the bipolar cell will synapse with the dendrites of the ganglion cell, forming the inner plexiform layer. Then we have the ganglion cell. These cells, axons of these cells will form the fibers will bend at right angle, starting to form the optic nerve. And then we have the inner limiting membrane. Again, dear students, the pigmented cell layer, rods and cones, outer limiting membrane, outer nuclear, outer plexiform, inner nuclear, inner plexiform, ganglion cell layer, layer of the optic nerve fibers, and the inner limiting membrane. Here, the best area of visual acuity known as the fovea centralis, reduction in the numbers of layers of the retina, and the photoreceptors are cones only. Fovea centralis. So the eye, we're going to have two slides, the eyelid, the cornea, and the retina. Regarding the ear, we're going to study the organ of Corti. The organ of Corti. You know that the ear is formed of external ear, middle ear, inner ear. The inner ear we have bony labyrinth and membranous labyrinth. From the bony labyrinth, we're going to study the cochlea. In the cochlea, there is the membranous labyrinth, which is the organ of Corti. This is a diagram showing the organ of Corti formed by the basilar membrane. The basilar membrane stand on it supporting cells and hair cells or sensory cells. These supporting cells, we have their name are pillar, phalangeal, Hansen, cladius, and border cell. All these are supporting cells. The sensory cells, we have inner hair cell and outer hair cell. First of all, we should know which is the outer and which is the inner. The outer, the hair cells are in 3 to 4 rows. So if you see 3 rows, so it is the outer. Here is the outer and here is the inner. So this is outer pillar, outer phalangeal, Hansen and cladius, inner pillar, inner phalangeal, and border cell. The hair cell ends with stereocilia that's embedded at the tectorial membrane. Again, dear students, this is the basilar membrane or the basilar membrane. Stand on it the organ of Corti, where first I can see here 3 layers, 3 rows. So here is outer and here is inner. So this outer pillar, outer phalangeal, cladius and Hansen cell. Here is the inner pillar, inner phalangeal, okay, and here is the border cell. This is the tectorial membrane. This is part of the vestibular membrane. Again, dear students, here is the basilar membrane and here is the vestibular membrane. Dividing here the spaces. Here is scala vestibuli, here is the scala media containing the organ of Corti, and here is the scala tympani. This area contains the endolymph and those the perilymph, and this is the stria vascularis. I'm standing on the basilar membrane. Here outer 3 rows, so this is outer hair cell standing on the outer phalangeal, and then we have cladius and Hansen cell. While here is the inner pillar, inner hair cell, border cell, and this is the tectorial membrane. Now we start the trial exam, dear students. Here, dear students, identify the section. It is the eyelid. Why? From one side skin, from the other side mucous membrane, between the muscle and gland. Look here at the junction between, or the margin between the epithelium of the skin and the mucous membrane. I can see here a section of the hair follicle, which are the eyelashes, so it is eyelid. The red arrow points to the meibomian gland present in the torsus. The blue arrow points to the skin. The black arrow points to the skeletal muscle, which is orbicularis oculi. Again, dear students, this is a section in the eyelid. Number 1 is the skin. Number 2, the meibomian gland present in the torsus. Number 3, the eyelash. Is very nice photo here. It is LS of the eyelash. Number 4 is the muscle orbicularis oculi, skeletal muscle. Here, dear students, is the cornea. Very nice photo here. Identify the structure. The cornea, the membrane points by the red arrow, Bowman's membrane, while the green arrow, desmids. The blue arrow points to the keratocyte, while the star is the substantia propria. Again, dear students, this is the cornea. The green line is the substantia propria. The yellow arrow, non-keratinized stratified squamous epithelium. The red arrow, the desmids membrane. The blue arrow, the endothelium. Here, dear students, is the retina. Here is the sclera choroid, okay? And this is the layers of the retina. The black arrow, the pigmented layer. The black arrowhead is the outer limiting membrane. A, outer nuclear layer. B, inner nuclear layer. The rectangle is the ganglion cell layer. Okay. Here is the area of the best visual acuity, which is known as the fovea centrals. The blue line is the retina, while the red circle is the fovea centrals. The yellow arrow, the photoreceptors, while the green star is the outer nuclear layer. Here, dear students, I can see here the part of the cochlea. The arrow, the black arrow, is the basilar membrane. The arrowhead, the vestibular membrane, and this curved arrow is the striae vascularis. Okay, so number 1 will be the scala vestibuli, and number 2 scala media, and number 3 is the scala tympani. Here again, dear students, here is the organ of Corti. Number 1, I'm standing on the basilar membrane. In order to know where the outer, where the inner, look at here the rows of the hair cell. I can see 3 rows here, so this is outer. I'm going to start with number 1 is the basilar membrane. Number 2 is the outer phalangeal cells. Number 3 inner phalangeal cell. Number 4, outer hair cell. Number 5, inner hair cell. Number 6, outer pillar cell. 7, inner pillar cell. 8, the tunnel of Corti. Okay, and this is the pictorial membrane. Again, dear students, what is this? It's the organ of Corti. What is this? Is the basilar membrane, and this is the vestibular membrane. So this will be the scala vestibuli. Number 1 will be the scala media or the cochlear duct. Number 2, the scala tympani. Which is which is outer and which is inner? Look at here these rows. So here is the outer. The black arrow points to the outer hair cell, while the green outer phalangeal. Okay, and this is the outer pillar, inner hair cell, pictorial membrane. Again, dear students, this is the organ of Corti. Here is the vestibular membrane, and this is the basilar membrane. Here is the red star, scala vestibuli. Here is scala tympani, and here is the scala media of the cochlear duct, which is out, and this is the stria vascularis, which is outer, which is inner. Look here, I can see the 3 rows of cells, so this is outer and this is inner. The green arrow points to the pictorial membrane. About the label diagrams, here we have the eyelid, section covered by skin, keratinized stratified squamous epithelium with hair follicles, sebaceous glands, glands of moles and Zeiss. The other side, palpebral conjunctiva stratified columnar epithelium with goblet cell. At the margin, there is eyelashes. Here I'm having dense connective tissue, which is the tarsus embedded at it, the meibomian gland. While here is the muscle, skeletal muscle, which is the orbicularis, oculi, and levator palpebrae superioris. Here is the cornea. It's formed of epithelium, non-keratinized stratified squamous epithelium resting on Bowman's membrane, substantia propria formed of regular collagen bundles in between the keratocytes, Descemet's membrane, and the endothelium. Here is the retina, pigmented epithelium, rods and cones, outer limiting membrane, outer nuclear, outer plexiform, inner nuclear formed of cells, the bipolar, horizontal amacrine molar cell, inner plexiform layer, then the ganglion cell, then the nerve fiber, and the inner limiting membrane. Last photo is for the organ of Corti. Here is the basilar membrane. Here is the vestibular membrane. Here is the pictorial membrane. From where outer or inner, look at the rows. In the row, if I having 3 to 4 rows, so it is outer hair cell standing on the outer phalangeal, inner hair cell standing in the inner phalangeal, near the inner phalangeal supporting cells known as border cell. Near the outer phalangeal we have Hansen and Clady cells are also supporting cells. The hair cell and vestibulocelia that embedded at the pictorial membrane. Thank you, dear students.